Now, gene transfer, this is interesting. We've already shown how bacteria multiply. They grow and divide by binary fission. So if you remember that animated slide, we had one bacteria that became two that became four. There's not a lot of space for changes to the genome. You do have mutations arise, but again, that's not frequent enough to compensate for all of the different varieties of bacteria we have as far as virulence factors, as far as resistance to antibiotics. So new combinations are generated in bacteria by the three main mechanisms we're getting ready to talk about: conjugation, transformation, and transduction. We're going to talk about transformation first because it's the easiest. We want to start easy, we'll hook you in, and then we'll get to the most complicated really, which is probably transduction. Transformation is like shopping for DNA, okay? You're sitting around and all of a sudden you see some strands of DNA. Under certain circumstances, if you're a bacterial cell, we might say you're competent to receive those strands, which means that you can actually transport them across the membrane and get them now into the cytoplasm. The only way that they can be expressed is through the process of homologous recombination. So we've taken up a linear piece of DNA. We had some cells maybe right around us that died. We got chopped up pieces of DNA available now, and we want a new piece of DNA. So we become competent where we're physiologically capable of carrying that strand across the membrane, and now in order for it to be expressed, it has to find a region of the chromosome that it's homologous to and then exchange by homologous recombination. Now this is an animation that I've done to help you understand how transformation might work. So this is a bacterial cell. This is going to be the cell that takes up the DNA. This is going to be the chromosomal DNA. So what happens is you have linear pieces of DNA coming in as identified by those red pieces of DNA. Again, under certain physiologic conditions, linear pieces of DNA can be taken up and internalized by bacterial cells. And when that happens, we say that the bacterial cell has become competent to do that. So in a competent bacterial cell can take up linear pieces of DNA, and now all of these pieces of DNA with this little white dot representing Rec A are looking for areas of homology on that bacterial chromosome. If they find areas of homology, they then exchange with similar alleles on the chromosome, and you get an exchange of genetic information. Okay? So that's transformation, and the best example to make this clinically relevant I can give you for transformation is going back to chapter 2 and what we talked about for Streptococcus pneumoniae being the most important virulence factor. We said the capsule was the most important virulence factor for strep pneumo. So uncapsulated strep pneumo was hanging out one day and said, hey, this piece of DNA looks interesting. What's a capsule? I don't know. But it was competent. It took it up. And now it went from just a non-virulent strain of Streptococcus pneumoniae to a very clinically relevant pathogen. So Streptococcus pneumoniae acquired its capsule through this process of transformation. So that makes that process, gives you a little bit of clinical relevance for transformation. The next mechanism of gene transfer that we're going to talk about is conjugation. Conjugation requires direct cell-cell contact from one bacteria to another to allow the exchange of genetic information. Now, what you need for conjugation, we're going to talk about some terminology and some rules. You need something called a fertility factor, and we'll talk about what that is. We are now going to introduce the sex pili. We talked about virulence pili that help organisms adhere to surfaces like E. coli in the urethra, for example. Now we're going to talk about the sex pili, which is basically a nice little tunnel made from one bacteria to another that allows it to exchange genetic information. Now, terminology-wise with conjugation is actually kind of humorous because it's the closest bacteria get really to mating here with conjugation. So in this case, we have a male cell that's actually the donor of this plasmid or DNA, and the recipient of this is the female cell. So we're talking male cells and female cells. Male cells are called F+ cells because they have the fertility factor, and female cells are called F- cells. All right? So the donors of this entire gene transfer are the F+ or male cells, and the recipients are the female cells or the F- cells. Now, there is a 50-50 shot here with conjugation. If we're talking about transferring a plasmid, then we don't require homologous recombination. However, if we get random bacterial genes introduced by conjugation, and I'll show you an example of that, that would require homologous recombination in order for them to be expressed. Okay? So let's look at what the donor cell has that the female cell does not have. All right, this is known as the F factor, and the F factor encodes for fertility genes such as the sex pili, very important, remember for the cell-cell contact, encodes for genes that stabilize the pair, allow them to stay together long enough to exchange that genetic information, genes that direct the transfer. And here we're really talking about the ORIT, the origin of transfer, okay? And you can also, in some plasmids, have an insertion sequence in which the plasmid can actually integrate into the bacterial chromosome. So this is what a fertility factor might look like at its most basic form, okay? So we would call a cell that had this fertility factor, we would call it an F+ cell. Now, I'm going to give you some examples of the cells we're going to talk about for these different conjugal crosses, okay? Female cells, we talked about male cells, these are the F+ cells. Female cells are called F-, okay? And they do not have the fertility plasmid. Now, there are two types of crosses that you can have, but in every cross, the recipient has to be an F- cell, okay? So we're going to talk about conjugation between an F+ and an F- cell, and then we're going to talk about conjugation between what we call an HFR cell and an F- cell. In each case, there has to be a recipient or a female cell in the mix. Now, the HFR strain, you can see, has this plasmid integrated into the bacterial chromosome. So HFR stands for high frequency of recombination. High frequency of recombination, HFR. So those are the crosses we're going to be looking at.